So yeah, as I guess Jesse introduced, the projects that I was working on at St. Jude Children's Research Hospital basically focused on proteins that were intrinsically disordered, that lacked a stable, stable three-dimensional structure. And what we were, I guess, fundamentally interested in was both how changes in the environment could affect the conformation dynamics of individual proteins, and how this can then be kind of overlaid onto the higher order assembly of these proteins into kind of oligomers and then eventually phase separated states. So to start this talk, I want to kind of give a bit of background as to why we were interested in this to begin with. And this kind of fed out of the emerging field of membraneless organelles or biomolecular condensates that kind of came to prominence around, I guess, maybe about a decade ago, but really about 2015. And what I'm showing here is kind of a cartoon image of a neuron that was published in a review in Nature last year, basically listing everything that has been identified as a membraneless organelle. So there's just essentially a wide array of structures within a cell that seem to come together and form sort of punctate structures in the absence of any containing membrane. So our work was really kind of focused on how we can understand the physics of this assembly from looking at minimal systems. So I guess the overriding principle of the work at BioCAT was asking the question of whether we can kind of infer mesoscale properties by looking at model systems. And kind of I've displayed this here in cartoon sense, like looking at a single punctate structure here, which in this case is a stress granule. If you zoom in on it, you can see there's a wide variety of kind of disordered proteins, nucleic acids, folded proteins. You zoom in further, and essentially what you're looking at are these small scale one-to-one multivalent interactions between subregions of individual disordered systems. So I think I like to use this example to highlight the utility of looking at model systems to understand kind of the complex nature of these biomolecular condensates. And the nuclear pore complex kind of is the archetypal membraneless assembly, or archetypal kind of phase separated complex. So while it's kind of restrained in two dimensions by sort of the structure around the nuclear pore, the interior of the nuclear pore is largely unconstrained, and the structure is generated by kind of phase separation and gelation of a series of phenylalanine rich disordered proteins. And the reason why I think that this particular system is of interest to us is that it's been reconstituted in vitro using very minimal systems. So essentially here, gel has just been made using one protein, NUP98, which is one of these phenylalanine glycine rich proteins. And what's really fascinating is you can see that the selectivity of the nuclear pore is more or less recapitulated just with these minimal one component systems. So it kind of highlights the fact that a lot can be learned by looking at kind of reduced systems and understanding the physics there. So in this talk, I'm going to kind of give a brief overview of what we have done and of looking at our model systems, particularly focusing on the work at BioCAT. And I kind of want to isolate this into two essential sections. The first is looking at the properties of individual proteins and basically looking at the notion that if you can very precisely measure what's going on with single proteins, you can kind of infer what's going to go on in mesoscale systems. And then second, after that, I'm going to try and link these single protein concepts into sort of a larger emergent system and see what we can talk about looking at the thermodynamics of phase separation and its kinetics. So the model system that we worked at and worked with at St. Jude Children's Research Hospital was an RNA binding protein called HNRMP-A1. And this protein kind of came to our attention again, like sort of in 2015 at the dawn of the biomolecular condensate field, because it was found mutations in this protein were found to be significant in ALS, and it was found to be enriched in these condensate stress granules. So work in Tanya Mittag's lab in collaboration with Paul Taylor's lab in 2015 dissected this protein and realized that the RNA binding domain was kind of dispensable for its localization to stress granules and its sort of assembly into phase-separated droplets in vitro. But the disordered low complexity domain at the C-terminus of the protein could itself form droplets in vitro. So essentially, the model system that we look at is kind of the isolated, disordered, low complexity domain of the C-terminus of HNRMPA1 as kind of a vehicle to examine the physical properties of phase-separated systems. So to delve a little deeper into what we mean by disordered low complexity domain, I just want to give a bit of a background of what the sequence features of this system are. So what I'm showing here is just the sequence of the disordered region of HNRMPA1. What you can tell is that this is basically the second half or the final half of the sequence. It's shown disordered here. And I think it's really highlighting what we mean by low complexity domain if you just sort this sequence by amino acid or amino acid type and realize that the vast majority of this sequence is glycine or small polar amino acids. There's this smattering of aromatic amino acids and some charged amino acids. So I think we basically were dissecting this for years, and a large number of experiments went into this. But to make the long story short, I'll just go back to kind of the unsorted sequence and say that we began focusing on the aromatic amino acids in the sequence, because if you kind of naively look at this by eye, you can see the black labeled sequences are kind of evenly punctuated throughout the sequence. And it seemed weird that we'd have a bunch of hydrophobic amino acids in a disordered region. It seemed weird that they were evenly spaced. So I think what we were trying to kind of get at is very well characterized by a series of molecular dynamic simulations that we ran on the low complexity region. And what I'm showing here is just kind of a analyzed result from one of these simulations showing essentially what we had theorized occurring in the simulation. So what is shown here is basically all of the aromatic residues dotted in yellow and looking at contact probability between, or I guess in this case, normalized distance between different regions in the protein. So what we see is that when you see a cross peak between any one of the aromatic amino acids, those regions of the protein are closer together than they statistically would be at random. And we can represent this kind of in this network diagram you see on the left and show that there's all these kind of network connections or contacts between the aromatic amino acids. So I guess the idea that we were coming to from this was that all of these sort of aromatic amino acids are forming transient contacts with each other, and they form a series of kind of weak, attractive, multivalent contacts that could eventually lead to the assembly of HNRMPA1 to phase-separated structures. So to kind of get a bit deeper into what we mean by this, essentially, we're going to take this information or what we've looked at on the single protein level and see if we can map this onto emergent properties. So the first thing that we kind of think about, particularly with respect to X-ray scattering, is what's happening when you modulate contacts within the protein. And the most obvious thing that could happen to a disordered protein is if there are very few contacts interchain, like if it's not contenting itself, you see a very large radius and a very large apparent scaling exponent. As you kind of increase, and kind of going back to the left, increase the number of contacts within the protein, you start kind of collapsing the protein down. So you see this sort of continuous collapse into sort of more collapsed structures. So essentially, you can kind of imagine the fact that if you have more adhesive regions of the protein, you're going to have a more compact structure. If it's more repulsive or they aren't interacting, you'll have a more extended structure. So with that in mind, we set about to kind of experimentally characterize HNRMPA1. And okay, so I guess included this here as well. So the way that then we kind of map this onto sort of emergent properties is to think about it in terms of a phase diagram. So if you can imagine a single protein going from extended here to collapsed here, the phase diagram essentially represents many proteins going from disperse into kind of collapsed into droplets. And this is characterized by the phase diagram, which is sort of the thermodynamic currency of phase separation. So here we have kind of a two-axis system, temperature and protein concentration. And we're looking at kind of moving the system horizontally across here from region 1, where you can think of proteins being dispersed or being kind of very extended, into a kind of the region of the phase temperature concentration region of the phase diagram, where you kind of would have more collapse, and the system is collapsing into kind of droplets. So experimentally, the way that we looked at this originally was just simply with equilibrium size exclusion coupled X-ray scattering measurements. I don't know, I imagine these have been covered in depth in this course, or at least they will be. And the way that we, I guess, modeled this system was by kind of treating these disordered proteins as though they were kind of random polymers, or imagining them kind of in the same lens that people in polymer physics or polymer chemistry would do. So what I'm showing here is the equilibrium data for the low complexity domain of HNRMPA1, the wild type data. And I'm comparing it here between two kind of extremes. This is a polymer that's a self-avoiding random walk, and this is sort of the classic scattering profile of a sphere. So from this data, we can kind of fit this to a model in which we can extract these two relevant parameters, the radius, which we see is, I guess, 27 angstroms in this case, and our scaling exponent, which is 0.42, 0.44. But importantly, we can kind of start getting a picture of what's going on with this protein, because the native state of it is more compact than this random walk where the protein has no adhesive interactions. It's more extended in a sphere, so it kind of lives in this middle ground where it's kind of sticky, but not completely. So we kind of transition from the single protein and start mapping in the phase diagram that I just mentioned before, so we can kind of map temperature concentration space and look at the boundary region between the one phase and the two phase regime. So we see that this fits fairly well to a mean field model. What we can also do is analyze this with respect to simulations. So we can parameterize coarse grain simulations that match these single chain dimensions, and then look to see how that simulation phase separates. And again, we can see that this matches our data fairly well, which gives us a good confidence that what we're seeing in the single chain situation maps pretty well to the emergent properties of the system. So to kind of get a bit deeper into this and kind of confirm that our model is correct, we started making variations of the wild type low complexity domain. So first, we kind of increased the number of aromatic amino acids by 30%, and then made a series of deletions where we essentially removed aromatic amino acids. And if I guess our hypothesis was correct that these are kind of the dispersed interactions, we should seek a fairly linear trend between the number of aromatic amino acids and the shape of the protein. And indeed, this is what we observed. So here we're again just showing the equilibrium measurements. And as you increase the number of aromatic amino acids, you see this kind of collapse of the protein. Removing aromatic amino acids results in kind of expansion of the protein. And this trend is quite predictable, relating the radius or scaling exponent to the number of phenylalanines and tyrosines. So I guess the million-dollar question is this still couple to the model that we created for kind of predicting the emergent properties? So here again, I guess we return back to our coarse-grained model, and here's the wild type that we looked at before. Our coarse-grained model did a pretty good job of predicting the radius of gyration of both the all of the aromatic variants, and remarkably did a particularly good job predicting the boundaries of the phase diagram for both the aromatic added and aromatic removed variants. I think interestingly here, the model predicted that this double minus variant wouldn't phase separate at any accessible temperatures, which was completely in line with what we saw experimentally. So I think this sort of bit of the talk kind of serves as an introduction to the way that we were thinking about the system, that kind of characterizing the thermodynamics of assembly of the low complexity domain into phase separated compartments. But we kind of wanted to delve a little bit deeper, because generally the hypothesis in cell biology is that one of the reasons why these membraneless compartments are so ubiquitous without life is because they have the ability to rapidly respond to changes in the environment. And what I am, I guess, showing here is a protein YAP1 that I guess exemplifies this quite well, that responds to osmotic stress by forming kind of rapidly forming punctate structures. When you remove the osmotic stress, the punctate structures disappear. And schematically, with respect to a phase diagram, we can kind of see what's going on here. You're kind of moving across this phase boundary. And so what we set out to do with kind of the kinetic measurements that are possible at BioCAT was to map exactly what happens to a protein as it crosses this phase boundary, to see kind of what the kinetics, what the response of the system is to a rapid perturbation from the environment. So the theory around phase separation suggests that there should be a nucleation event. Essentially, what this means schematically is that when you're in kind of concentration regime 1 here outside of the two-phase regime, there's kind of an exponentially increasing free energy cost to forming clusters of molecules. As you move exactly to the phase boundary, this cost becomes a bit shallower, but there's still kind of a monotonically increasing cost to increasing clusters of proteins. And then once you cross into the two-phase regime, the curve begins to develop this nucleation barrier. So essentially, there is a point where the favorable energy of entering a cluster outweighs the kind of surface energy cost of that cluster forming. And this energy here is sort of characterized as the kinetics of how quickly a system is going to kind of enter the phase separated regime, or enter the two-phase regime, after you rapidly perturb the system into kind of this region of the phase diagram. So this experiment that we basically use as the bread and butter of this was alluded to by Jesse in the previous talk, which was the phase separation of the low complexity domain that we were looking at can be induced by increasing the sodium chloride concentration. And you can map a phase diagram that it looks very similar to the temperature phase diagram as a function of sodium chloride concentration. And you can use this as sort of your, I guess, thermodynamic currency to probe the kinetics of this reaction. So essentially, we're going to rapidly quench by adding sodium chloride from the one-phase regime into the two-phase regime. So if we back up a bit to the first section of this talk, we were looking at the collapse of single proteins as a proxy for how they were going to phase separate. And if this is indeed sort of a good proxy, then essentially we should be able to observe this at very fast timescales using the chaotic flow mixers that Jesse had previously alluded to. And we were honestly sort of a bit surprised that we were able to see this quite readily. So what we're looking at here is just a trace of the radius of gyration as a function of time after mixing. And this is sort of our wild type phase separation competent low complexity domain. You see this kind of very rapid collapse at the beginning of the mixing period, and this kind of rescue as kind of clusters of molecules begin forming. And interestingly, we saw kind of basically no salt sensitivity to the aromatic depleted low complexity domain. So we know that in this construct with very few aromatic amino acids, there's no phase separation. There's also no salt sensitivity. To kind of make a brief detour into how we looked at this data, we also observed this kind of interesting phenomenon here, which was an increase in the zero angle scattering that was coupled to the decrease in the radius of gyration. And in principle, this was kind of a illogical conclusion that you would, if anything, expect a decrease in the zero angle scattering as you saw a decrease in radius of gyration, a slight decrease. So we set out to kind of hypothesize why this could possibly be the case. And what we eventually landed upon was a theory where you have differential hydration between a kind of collapsed disordered protein and an expanded disordered protein. And essentially what this means is that when you're collapsed, you've either trapped hydration waters, or the hydration waters are kind of less dynamic than they would have been previously. So we kind of had limited data to prove whether or not this was existing, but we set out to sort of model whether this was possible. And we modeled this in two separate ways. The first was kind of a toy model where we were looking at a core shell system, where essentially you're considering your disordered protein as a core and your hydration layer as a shell, and the differences in contrast between the two of them could be a proxy for how stable the hydration scale is. So essentially, as you map the excess core density, you see the exact trend that you would see in this particular model. So you see a decrease in radius coupled to an increase in zero angle scattering, suggesting that this kind of trend in the data could be possible. And we can do this in another way, which is, I guess, either more sophisticated or less sophisticated, depending on how you look at it. And this is essentially calculating what the scattering profile should be for a series of conformations of the disordered protein, and basically toying with or adjusting what the scattering density of the coupled hydration shell is in these proteins. So we did this with a series of different conditions, essentially low contrast, high contrast, and then what we had expected to happen in this interesting case where if you collapse the protein, you have a more stable system, where we have kind of high contrast and compact conformations and low contrast and extended conformations. And again, we see this trend where you decrease the radius and increase the zero angle scattering. So this was just kind of an aside, and I think I'd stress to anyone that this was a kind of speculative, or just kind of using modeling to see how we could predict the trends in the data. And with a lot of X-ray scattering data to kind of be conclusive, I think other measurements would probably be necessary. So I think it was really cool that we could see this sort of collapse, the single chain immediately after we quenched into conditions where we should phase separate. But we really wanted to see what the kinetics of the formation of the phase separated system were. So for this, we again switched to sort of the longer timescale mixer that Jesse was talking about, the laminar flow mixer. And we're essentially watching the formation of microstructures of dense phase protein. And what this experiment essentially relies on is that the dilute phase protein and the dense phase protein have fundamentally different scattering profiles. So we can essentially use the laminar flow mixer that Jesse discussed and watch the evolution of this sort of zero time point system into kind of the later time point scattering profile. And in order to analyze this data, we essentially kind of extracted an assembly metric, which was how far the low angle scattering intensities were departing from what you would expect from sort of an ideal dispersed system. So assembly at 0 times is small, at later times you see a much larger assembly metric. So you watch the data sort of as a function of time. You see the evolution of kind of the assembled states. You calculate the assembly metric, and you see this sort of classic plateauing curve that you would expect from a nucleated event. So to analyze or to kind of delve into the thermodynamics of this system, we essentially titrated the concentration of salt that we were mixing our protein into. And what this titration should do is kind of what I'm mapping here, is moving the point closer and closer to this phase boundary. So as you decrease the salt concentration, you get closer to the phase boundary, and essentially that nucleation barrier, or free energy of forming clusters of molecules, should, I guess, get larger the closer to the barrier you are. So how this manifests in the data that we're looking at is at high salt, you see sort of a very rapid nucleation and formation of phase-separated structures. As you decrease the salt concentration, this gets slower and slower. And interestingly, when you get very close to the phase boundary, where I was suggesting you still have kind of these near monotonic barriers to nucleation, we see essentially no formation of phase-separated structures on the time scale of the experiment, which is, I guess, completely in line with what we expected. We can kind of map this to the, I guess, theoretical expectation of, I guess, homogeneous nucleated event, which is that essentially there should be a well-defined relationship between the rate of nucleation and, I guess I'm showing the supersaturation here, which is essentially how far the system is inside of that two-phase regime. And again, we can kind of predict quite well the trends we observed based on the data that we recorded. So I think I have one final note to make before I wrap up this talk is that the data that we extracted from the time-resolved X-ray scattering experiments showed quite well kind of that this was on certain length scales of homogeneous nucleation event, described quite well by the models that we consider for this. However, in all cases, we observe this second region, or sort of this lag phase, the beginning of the curve, which is not predicted by kind of a homogeneous nucleation model. So we wanted to kind of look deeper into what was going on at these sort of time points here, the very beginning of nucleation, and see if we could get a bit more information. And I guess, as I mentioned earlier, that with a lot of X-ray scattering experiments, it's very good to look at complementary methods. And in this case, we used analytical ultracentrifugation to sort of model what was going on very close to the phase boundary with respect to the formation of oligomers. So what I'm showing here is sort of the analytical ultracentrifugation data remodeled into kind of a distribution as a function of sedimentation coefficient. And we can see that at 50 millimolar salt, we have kind of a formation of some kind of higher order oligomeric structure. So we can kind of do a titration and see the formation of these oligomers as a function of protein concentration, and model this to a kind of pseudo-isodesmic model. So the reason I say pseudo-isodesmic model is that sort of an isodesmic assembly would assume that every protein molecule is added to the cluster with the same affinity and the same dissociation constant, which is exactly how you would expect assembly to occur, or clusters to form in a homogeneous nucleation model. You would expect that you see fluctuations in density of the system as individual molecules are added or subtracted from the clusters. But instead, we have to model this with kind of a two-state model, which suggests that there is a barrier to original oligomerization forming, original oligomers forming, after which you can start adding additional oligomers with this kind of similar KD that you'd expect from homogeneous nucleation theory. So I guess in conclusion, I kind of want to wrap this up with this pseudo reaction coordinate to describe essentially what we were talking about with the kinetics, which is that kind of you go along this coordinate, your individual molecules have one barrier in which they form kind of initial small oligomers in the solution. And then you further encounter a second barrier, which is your nucleation barrier that's salt sensitive, that can eventually be on pathway to phase separation. So I think I'm about out of time. So I'm going to go ahead and just thank everyone involved. This work was largely done in Tanya Middag's lab at St. Jude Children's Research Hospital. along the way, all of these people contributed. Of course, at Argonne National Lab, Jesse and Srinivas were critical to this work, along with kind of all of our external collaborators as well. And happy to answer any questions that anyone has.